In these sections, what we're going to do is start talking about the actual ways in which these molecules begin to build some of these upper level structures for us, and look at some of the kinds of interactions it'll take as we do this assembly of molecules. And then the next lecture we'll talk a little bit more about the nature of some of the ways in which these molecules will interact with each other. So at the chemical interaction level, this is kind of like your chemistry of life material that you would have had in a course before. I think if you basically recall back to the material you would have had at the 105 level, for example, remember that about 28% of the chemical makeup of a cell is made out of macromolecules. And of course, what do you know about the other 72%? We know that about 70% of that is water, so that leaves us 2% of everything else. So these are a pretty important piece to the dry weight of a cell. We call it dry weight of the cell. If you were to take all the water out, what's left, well over 90% is made up of these things called macromolecules. Macromolecules, by definition, as you would expect, are large. They're polymers, as we've already discussed, and they tend to be built out of these same types of building blocks that are held together. So we'll get into some specifics about a couple of the macromolecules, but remember that the building blocks themselves are going to be held together by covalent bonds. And of course, if you need to go back and review some of your intro chemistry stuff, covalent bonds are those bonds where two atoms in a molecule are actually sharing an electron in one of their valence shells. It's a very stable, almost permanent interaction between those atoms in that molecule, and really there would have to be something that is going to injure or introduce that damage to these molecules. Or an enzyme could actually break a covalent bond too, of course, if it's a bond we want to break in those cases. Now, as far as the macromolecules go, remember there's 4 different types. We've got the carbohydrates and the lipids. They tend to fulfill structural roles and energy a lot, although we'll also see some communication aspects here. And we'll talk about some structural roles as well as we get into some of the organelles. As you can guess, the plasma membrane is going to involve a lot of lipids, so we'll talk about those there. The other two we're going to spend quite a bit more time on, simply because the nucleic acids are where the gene information is stored. We already talked about how you have these instructions on how to build things in those genes. The nucleic acids would be a primary mechanism by which that is going to happen. And of course, the information that those nucleic acids carry is how do we go about building a protein in order to do something. So these proteins play a lot of different roles. They're a major contributor to the metabolism inside of a cell. So they're not just enzymes, but they're also going to be structural. They do transport, they do protection, they provide energy. And so there's just a lot of things that proteins can do. And so certainly these are not last because they're the less important. They're last because they're probably the most important. Once they're built, these macromolecules are going to interact with each other through what you guys would have learned as non-covalent bonds. Remember, there were others in your chemistry class that you probably worked on, like van der Waals forces and others, but these 3 are going to be the ones that primarily are going to play in the conversations that we have. So definitely spend some time reviewing in your mind what these are. The hydrogen bonds, of course, are going to be 2 electronegative atoms, like a nitrogen or an oxygen. And so if you have something like an O or an N, remember these guys are really electronegative. And electronegative means that these guys are greedy for electrons. So if this is a covalent bond and these guys are holding it, that means that this pair is going to be closer to the oxygen, which leaves this guy out here with a slight positive. This guy, if he's in a molecule, he's going to pull those electrons closer to him, okay? And so that, of course, gives him a negative charge. That's a little delta sign. And so the question would be then, or of a hydrogen bond, would be these guys, where this slight negative and that slight positive is going to interact between them. Okay, so that would be what's called a hydrogen bond. Ionic bond, of course, one of your probably best examples of that would be a sodium and a chloride, where an entire electron gets transferred over. This guy takes on a negative, this guy takes on a positive, and those guys will interact. And then, of course, hydrophobic would be things that are the opposite up here. So these guys are polar, because you end up with two poles across that bond, but something that does not, or basically shares things equally. So carbon is not going to be very electronegative. So in this case, those electrons are going to be right in the middle. And what that means is that these guys then tend to be hydrophobic, because remember, water, what does water look like? H2O is very polar. Okay, and so very polar means that I'm going to like to interact with polar over here. So this is what you might find in an aqueous environment. This is what we call nonpolar. And so this does not like the water environment. So these here are going to be it's not so much an interaction as it's just these guys are going to like to be with each other more than they're going to like to be in the aqueous environment. So hydrophobic interactions will be where these hydrophobic or non-polar parts of a molecule tend to fold inside where the aqueous parts of the cell can't get to it. Okay. Each one of these individually pretty weak, but if you combine them all together, you will end up with interactions that are, one, very stable, but of course the other thing we don't want this to be is permanent. We want to be able to take these apart. So this is why non-covalent bonds are really important for biology, is because the way in which we go about interacting with each other needs to be a little bit transient. We need to come apart and interact with others in order to carry out things that are important for life. Okay, so here would be a good example of both covalent bonds and actually you find covalent bonds here with the molecules that are what's holding together, the bonds that are holding together the molecules themselves. These would be the nucleotides, G's, A's, T's, and C's. These, of course, you'll remember held together by hydrogen bonds. And of course over here you're going to have these negative charges with these phosphates, and that means that anything interacting with a backbone on DNA is going to probably interact with ionic bonds. Hydrophobic, not a whole lot to do with this guy, because remember, this is deoxyribonucleic acid, so it has a very negative charge, but we'll see hydrophobic interactions come into play as well as we move on forward. Okay. All right, so in the next section, I'll end this one here, but the next one, what we'll do is actually talk about how these particular bonds are used to assemble structures and some of the important features that we find within that assembly and things that cells can do to actually regulate that assembly, so that in very real time we can change who is interacting with whom, and we can control what kinds of interactions are taking place. They're not just random. Okay, so see you then.